Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that take place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On average, 1100 to 1200 milliliters of blood is filtered by the kidneys per minute. The glomerular capillary blood pressure causes filtration of blood through three layers: the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The epithelial cells of Bowman's capsule, called podocytes, are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma, except the proteins, pass on to the lumen of the Bowman's capsule. Therefore, it is considered a process of ultrafiltration. The amount of the filtrate formed by the kidneys per minute is called the glomerular filtration rate, or GFR. GFR in a healthy individual is approximately 125 milliliters per minute, which is 180 liters per day. The kidneys have built-in mechanisms for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by the juxtaglomerular apparatus, or JGA. A comparison of the volume of the filtrate formed per day, which is 180 liters per day, with that of the urine released, which is 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in different segments of the nephron perform this either by active or passive mechanisms. For example, substances like glucose, amino acids, sodium ions, and so on in the filtrate are reabsorbed actively, whereas the nitrogenous wastes are absorbed by passive transport. Reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like hydrogen ions, potassium ions, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of body fluids. Function of the tubules. Proximal convoluted tubule, or PCT. PCT is lined by simple cuboidal epithelium, which increases the surface area for reabsorption. Nearly all of the essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia, and potassium ions into the filtrate, and by absorption of bicarbonate from it. Henle's loop. Reabsorption is minimal in its ascending limb. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of the loop of Henle is permeable to water, but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water, but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Distal convoluted tubule, or DCT. Conditional reabsorption of sodium and water takes place in this segment. The DCT is also capable of reabsorption of bicarbonate and selective secretion of hydrogen and potassium ions, as well as ammonia, to maintain the pH and sodium-potassium balance in the blood. Collecting duct. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Large amounts of water can be reabsorbed from this region to produce a concentrated urine. This segment allows the passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of the blood by the selective secretion of hydrogen and potassium ions. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite directions and thus forms a countercurrent. The flow of blood through the two limbs of vasa recta is also in a countercurrent pattern. The proximity between the Henle's loop and vasa recta, as well as the countercurrent in them, helps in maintaining and increasing osmolarity towards the inner medullary interstitium. This gradient is mainly caused by sodium chloride and urea. Sodium chloride is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. Sodium chloride is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the countercurrent mechanism. This mechanism helps to maintain a concentration gradient in the medullary interstitium. The presence of such an interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate or urine. Human kidneys can produce urine nearly 4 times more concentrated than the initial filtrate formed. Regulation of kidney function. The functioning of the kidneys is efficiently monitored and regulated by hormonal feedback mechanisms involving the hypothalamus, JGA, and, to a certain extent, the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate these receptors, which stimulate the hypothalamus to release antidiuretic hormone, or ADH, also known as vasopressin, from the neurohypophysis. ADH facilitates water reabsorption from the latter parts of the tubule, thereby preventing diuresis, which is the increased or excessive production of urine. An increase in body fluid volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect kidney function by its constrictory effects on blood vessels, causing an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA plays a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, or GFR, can activate the JG cells to release renin, which converts angiotensinogen in blood to angiotensin I and further to angiotensin II. Angiotensin II, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. Angiotensin II also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium and water from the distal parts of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor, or ANF. ANF can cause vasodilation, or the dilation of blood vessels, and thereby decrease the blood pressure. The ANF mechanism, therefore, acts as a check on the renin-angiotensin mechanism.